Welcome to Micro Review. Today our topic of discussion is Clostridium Perfringens. It causes two diseases. One is gas gangrene and one is food poisoning. Up till now the bacteria we have studied, they are all causing single disease, but this particular bacteria is causing two diseases, gas gangrene and food poisoning, both of which are totally different from one another. If we talk about the features of this bacteria, as I mentioned earlier, that all the Clostridial species are having some things in common, like they're all anaerobic, spore forming, gram positive rods. If we talk about its habitat, so it is found in soil and it is the normal flora of colon as well as vagina. This is an important MCQ point. One thing to remember is that up till now we have studied Clostridium tetani, Clostridium botulinum, Clostridium perfringens. They all are present in soil. This is a thing to remember. Moving on to our topic of gas gangrene specifically. It is transmitted via the wounds. The wounds are not small wounds or minor wounds, they are actually major wounds, like the wounds associated with war wound, automobile or motorcycle accident, or any cases of septic abortions in which endometritis is occurring. If we talk about the wound more specifically, so it is a type of wound in which a large amount of flesh has been exposed to dirt or is contaminated. Pathogenesis begins with the traumatic wound. How is the gas gangrene being developed? The story begins with the traumatic wound, due to which vascular compromise will occur, due to which a favorable environment for anaerobic growth would be produced. When there will be anaerobic environment, Clostridium perfringens produces a toxin known as alpha toxin. Alpha toxin destroys the phospholipids in the cell membrane. It is actually a lecithinase that cleaves the cell membrane, due to which the muscle is destroyed, leading to myonecrosis, and RBCs are destroyed, leading to hemolysis. And the hemolysis it causes is actually double zone hemolysis. This is an important MCQ point, right? During this whole mechanism, the metabolism produces some gases, and these gases lead to the development of gas gangrene. So the question arises that what gases are produced and what gases are present in the gangrene. Those gases are carbon dioxide and hydrogen gas. Moving on to the clinical findings, pain is occurring, edema, cellulitis, gangrene and exudate. But it is not a normal exudate, it is an exudate that has now been mixed and tinged with blood. And it also leads to some systemic toxicity like hemolysis and jaundice. When there will be excessive breakdown of RBCs, it will lead to hemolysis, and the byproduct bilirubin would lead to jaundice. It also causes fever, hypotension and shock. But the main things to remember in the clinical findings is the gangrene development, the blood tinged exudate, the hemolysis and jaundice. Moving on to the clinical diagnosis, we see gas at the injury site on imaging and crepitus is felt. So what is crepitus? When we palpate that gangrene, we feel some gas bubbles in it. So this is known as crepitus. Bubbles of air in wound on palpation, mainly hydrogen gas. Moving on to the treatment. So the first line treatment, whenever a patient is coming with a major wound or any sort of automobile accident or traumatic wound, you have to do the surgical debridement, that is removal of dead, damaged and infected tissue. Then penicillin G, which is a broad spectrum antibiotic, is used for the treatment. You must note that there is no vaccine for this disease, due to which the mortality rate is high. If we talk about the prevention, it can be prevented by cleaning the wound and using penicillin for prophylaxis. If we talk about the lab diagnosis in case of gas gangrene, the bacteria is cultured anaerobically. As I mentioned earlier, there will be a double zone of hemolysis on blood agar. And if we want to detect the lecithinase, then egg yolk agar is used. And the enzyme identification is done by its inhibition with specific serum. And this brings us to the end of this topic. To summarize gas gangrene, you have to remember that it is transmitted via the wounds, major wounds, not a normal wound, major wounds, due to which vascular compromise is occurring, the area of avascular necrosis is present, due to which anaerobic environment has been developed. This anaerobic growth occurs, leading to the development of alpha toxin. Alpha toxin destroys the muscle tissue and the RBCs, leading to myonecrosis and hemolysis. During this whole mechanism, or we would say that during the metabolism of the toxin, it produces gases like CO2 and H2, which are present in the gangrene. Clinical findings are the pain, edema, blood-tinged exudate, gangrene has been developed, hemolysis and jaundice is occurring. These are the systemic signs. The local signs are pain, edema, cellulitis, gangrene, blood-tinged exudate, right? Diagnosis is mainly you feel crepitus. And the first line treatment is you have to surgically clean that wound, then you give antibiotics, and you have to note that there is no vaccine. That is why mortality rate is high. So you have to mainly focus for this particular disease on the prevention. So if you will be able to prevent it, you will be able to prevent the further spread. So you have to clean the wound and give penicillin for prophylaxis. Moving on to our second topic or second disease, which is food poisoning. You have a basic knowledge about food poisoning. So if we talk about the transmission of food poisoning, what happens is that contaminated food, which is containing the spores of Clostridium perfringens, is improperly cooked, due to which the heat resistant spores survive and they are ingested. This is how it is transmitted. From now on, this is the pathogenesis. After the ingestion, the bacteria is producing an enterotoxin, due to which diarrhea is occurring. Proper pathogenesis is this. Undercooked contaminated food, spores are ingested. Once the spores are ingested, the diarrhea occurs. So now the question arises that Clostridium perfringens is the normal flora of the colon. So why is it causing diarrhea? No doubt it is the normal flora of colon, but it is not the normal flora of small bowel, where it produces the toxin, which is enterotoxin, having a mechanism similar to superantigen, leading to food poisoning and diarrhea. The diarrhea is late onset diarrhea, which is mainly asked. So any sort of question that would be asked, that why is the diarrhea occurring late? Why is it late onset? Why is it occurring after 8 to 16 hours? So the answer is that Clostridium perfringens spores are ingested. The toxin is not ingested, as was the case of Bacillus cereus, where the food poisoning was occurring and the immediate effects were occurring. Here, the spores are ingested. And when the spores go in the GIT, they take some time to germinate, they take some time to produce the toxin, and the toxin takes some time to produce the effects, due to which this is the diarrhea of late onset, slow onset diarrhea or late onset diarrhea after 8 to 16 hours. Along with watery diarrhea, there are cramps and little vomiting. And you just have to give the symptomatic treatment. No specific treatment is given, only the symptomatic treatment is given, and it can be prevented by cooking the food properly, and there is no such lab diagnosis, right?